Agnes Flöhl, and we work on TDCS and aging, cognitive training, and I will also talk about computational modeling.
Florian.
Hi, yeah, my name is Florian Kasten. I'm a postdoctoral researcher at the University of Oldenburg, and I work with Professor Christoph Hermann, and we're mostly working on TACS and TACS in neuroimaging.
Good. Bernard.
Yes. Hello, everybody. My name is Bernard Sehm. I'm a neurologist, and I have a research group at the Max Planck Institute of Human Cognitive and Brain Sciences in Leipzig, Germany, where we investigate mechanisms subserving neurological recovery, motor recovery, and also motor learning.
Zeynep.
Hello, everyone. I'm Zeynep Esmailpour. I'm a PhD student with Mount Dixon at City College of New York.
Adam.
Hi, I'm Adam Woods. I'm an associate professor of clinical and health psychology at the University of Florida. We focus on looking at transcranial electrical stimulation, both in and in out, relative to treatment of age-related cognitive disorders.
Lucia.
Hi, everyone. Lovely to be here. I'm Lucia. I'm at Imperial College London. I'm a neurologist. I work mostly in multimodal imaging with TDCS and interested in individual variability, brain injury, rehabilitation, particularly in the cognition and psychiatry field.
Good. Catherine. Catherine, could you hear us?
Okay, sorry, I wasn't sure it was me. I'm Catherine Studley. I'm at American University. I'm an associate professor in psychology and neuroscience, and we focus on TDCS in a few different clinical disorders combined with functional imaging.
Okay, Joseph.
Hello, my name is Jose. I'm working in Nagoya Institute of Technology, Japan. We are working in electromagnetic computational modeling for TMS and TDCS, and also now we are working in nerve modeling. Thank you.
Good. Floel.
Yes, my name is Agnes Flöhl. I'm at the University of Greifswald in Germany, and I'm a neurologist, a neuroscientist interested in non-invasive brain stimulation, dementia, and stroke rehabilitation.
Yes, sir.
Yeah, I'm Gesa. I'm from the Max Planck Institute in Leipzig, and our group part here is working on cognition and plasticity, especially plastic effects of non-invasive brain stimulation.
Money.
Hi, my name is Irene Cabral Calderin, and as from next week, I will be a postdoc at the group of neural and environmental readings with Molly Henry at the MPI for Empirical Aesthetics. And I'm mainly interested in using TACS to modulate perception and using also brain imaging and computer modeling to improve our simulation paradigms.
Ghazaleh.
Hello everyone, my name is Aitana Saramani. I'm a student in biomedical engineering, and we are working on TDCS fMRI to find a good answer for inter and intra-individual variability in response to TDCS.
Good. Johannes.
Hello, I am Johannes Voskuhl. I am a postdoctoral researcher also in Christoph Hermann's lab. I did some work on TACS, fMRI, and recently we've been doing a project on the combination of TACS with MR spectroscopy.
Maro.
Hi, my name is Maron Dixon. Along with Zainab, I work at the City College of New York, and we've done a lot of work on computational modeling, and we are very interested in how to integrate that with functional imaging.
Alexander.
Hi, I'm Alex Opitz, assistant professor at the University of Minnesota. My lab works a lot, like say, computational modeling, also interested a lot now what happens in the brain during brain stimulation and physiology. And also, just wanted, if I can say that I'm also recruiting postdocs at the moment, pretty much about the topic of that seminar. So if anyone's interested, yeah, write me an email.
Great, awesome. So we still have others online, something like 33 attendees. so far. So if there is anybody else that we do not have his or her camera kind of online, he or she could introduce himself. Axel, would you introduce yourself as well? Axel, we cannot hear you. Your mic is not on, Axel.
Now better?
Yes, we can hear you really.
Wonderful. Yeah. Cool. So my name is Axel Thielscher. I'm working at the Danish Research Center for Magnetic Resonance and also at the Danish Technical University. I've done a lot of computational modeling with my group. We are also very interested in seeing how basically biophysical prediction, field prediction, linked to neurophysiological measures of TCS and TMS. So, yeah, and neuroimaging is obviously one of the possibilities to assess this, right?
Good. Look.
So.
Hello.
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 Hi.
Luke, would you?
Hi. I've just joined late.
Duke, could you introduce yourself?
Yes. Can you hear me?
Yes.
Hi. I'm Duke Shreen. I'm at the City University of New York, where I direct our MRI facility and have doing work with combining TDCS and fMRI in a safe and reliable manner.
Good. Makoto?
Hi, can you hear me?
Yes.
Okay. Hello, everyone. My name is Makoto Uji. I'm a postdoc research fellow in the School of Psychology and Neuroscience at the University of St. Andrews. I am interested in the neural oscillation and the cross-frequency couplings.
Great. Anybody else that would be kind of introducing via the mic?
Hello.
Hello.
Hi. Sorry, I just joined a little bit late. My name's Maria Ironside, and I'm at McLean Hospital, and I study TDCS and depression using behavioral and functional imaging.
Welcome, Maria. Anybody else who would like to introduce?
Yes, I'm also a little bit late. So my name is Daniel Kieser from Munich, from the University Hospital in Munich. And we are also doing a lot of multimodal and neuroimaging combined with TDCS.
Anybody else?
Hello, can you hear me?
Yes.
Hi, this is Aprenda. I'm a postdoc with Dr. Woods at University of Florida.
Anybody else? Okay, let's start. So today we are going to, basically, this is the second INTF webinar that we have. In the first INTF webinar, we discussed about challenges for doing TES fMRI studies, and we received lots of really nice feedbacks from people in terms of the good discussions that we had together. And we explored really great potential to see how we can do joint collaborations and how we can share protocols. So it seems that there are many interesting things that we can do together. So we are planning to do webinars every something like 3 months. So the next one would be in late September. It would be on, I think, the September 26th. So we will kind of inform you about the third one. In this one, in the second webinar, we are going to be mainly focused on the computational head modeling and functional brain imaging, how we can combine these two modalities together. And we know that we have variations in response in TES. I mean, those who are doing TES studies, they know that the variation is significant among different individuals, and even we have intra-individual variations. And the question is, okay, how we might be able to predict that intra-individual variation using different types of data that we have. And there are discussions in terms of what are the sources of these variations. And we know that potentially brain structure is one of the major sources of variation among different subjects and participants. And the question is, okay, how much computational health modeling could be a predictor for a good source of trait-based variation? And then we have also ideas in terms of, okay, it seems that brain function could also be a source of variation. And then we are exploring whether fMRI could be a tool to help us to get into that source of variation and see how we might be able to use that. And then the question would be, okay, how to combine computational model and fMRI? And there are lots of challenges in terms of, okay, how we are going to integrate data. What are the potentials for data fusion? What are the modeling things that we can do for that? And then there are challenges in terms of the scale of the integration and the space of integration. So there are many things that we will discuss today about. And then there might be interesting things in terms of how we might be able to collaborate. And is there any chance that we can share the pipelines that people in different labs they are doing in terms of the combination of computational health modeling and fMRI? And is there any potential for data sharing? And I've heard frequently from different colleagues who are doing TES fMRI, but they do not do computational health modeling, and they are interested to share the data that they have collected so far to see if we can combine that with computational health modeling. So I think that there are definitely interesting potentials for collaboration. So today we'll have different speakers from New York, from Denmark, from Germany, and I hope that these discussions would be a good starting point within the INTF network to establish a collaboration for future activities. But other than that, we are also moving forward to see how we might be able to put together a few consensus papers. And based on what we have been discussing from the last session and the last webinar, right now we are working with Duke and Ines in terms of how we might be able to provide a checklist for those who are interested to do online fMRI TES study, so doing TES inside the scan.
I think,
And
yeah,
just introduce the idea of what we are going to do and the things that we are going to kind of present in the checklist. Luke?
Yes. So we discussed last week this checklist, and we're in the stages of putting it together, but it would probably, some things that are very obvious to many in this webinar, but perhaps may not be obvious to people who would like to start TCS fMRI protocols. And so it's basically just a short checklist that we're going to expand upon that have to do with the alterations from your regular TDCS outside of the scanner setup versus in the scanner. Considerations about your electrodes, not to use sponges or saline solution, different recommendations for different types of gel, or options that you might have. We had a lot of discussion last time about initial experiences that many of us had shared, where our first scans were plagued with RF artifacts because of the way that the cabling was going through the waveguide, and the RF filters weren't really either weren't present or weren't doing the job. And so many of you mentioned having to learn the hard way to send the cabling through the penetration panel, not the waveguide, using appropriate RF filters, and how that solved a lot of the artifact issues. At the same time, I do remember, I don't recall who mentioned that might have been a difficult thing in Europe for safety compliance, sending the cabling through the penetration panel instead of the waveguide, and that might be a topic for further discussion or elaboration, because that was new news to me, and I think that's an important consideration. And as well, we would like to also grow this checklist out from just straightforward hardware considerations that are simple, but without having taken those precautions can result in large artifacts, to also more sort of advanced MR acquisition, design, and post-processing strategies to sort of further mitigate the potential artifacts in TDCS fMRI.
Awesome.
I welcome feedback from everybody.
That is great. So we will kind of start to share the initial draft with others who are doing TES fMRI. And it's interesting that we already have 54 published online TES fMRI study. And it would be interesting to see what people have done so far and what are the things that we might be able to consider in the future. And then, yeah, that would be nice to see how we can kind of move forward for that in a collaborative way. And meanwhile, Ines Violante, Dr. Violante, are you online right now? I cannot see you online. Ines is working on the other side of the checklist, which is basically about the cognitive neuroscience of the checklist. So Duke is mainly focused to the, let's say, physics part of the checklist, how we are going to do that. And Ines is working right now on the cognitive neuroscience part of that. Okay, what are the things that we can do when we put something inside the scanner, inside the combination of, let's say, fMRI task and stimulation? What are the things that we can consider, and what are the challenges that we will face in terms of doing the stimulation inside the scanner in terms of the cognitive neuroscience issue. So that will be another part of the checklist, and we'll just kind of share that checklist with you guys as well to see how your feedback. And we are hopeful to be able to put that out as something like an INTF consensus checklist to help people to kind of increase the quality of what they are doing and bringing potential for sharing the data together. That would be one of the things that we are moving forward for that. And meanwhile, we are hopeful to be able to have a sort of consensus paper from what we are discussing today in terms of the computational head modeling and fMRI. That would be another thing we'll discuss, and that would be really nice to see how those things would be happening. The good thing is that right now, Marom and Zainab, we are working on a paper that is hopefully will be published in terms of the integration. So they will discuss about what are the things that they have done so far and potentials for that integration. So let's go to the next speaker, Marom and Zainab. Marom, we are listening to your talk.
Great. Yeah, me and Zainab are going to give this together. Zainab, are you loading the slides up from your end, or?
Yeah, I do. But the thing is, how I can share my slides? Do I have?
Just we'll give you the.
Can I share my screen?
Yes, you will have that.
So while Zainab loads it up, I'll just give a quick introduction. So my lab has been doing, and Zainab has been doing also a lot of computational modeling of current flow. So prediction of how current flow flows through the head during TDCS. We're not going to talk about that today, but we and other, we've released open source software like Roche that allows everyone to do that. Axel and Alex, who are on the call today, released other software called SIDNIBs that allows everyone now to do current flow modeling. So we're not going to talk about that. We're actually going to focus on how to integrate these current flow models with functional imaging. And this is something we've been thinking about for a couple of years now. We've been working on this with a few people on this, just on this webinar here. We've been working with Hamed, Adam Woods, and also Duke, who's at my institution. And so what Zainab is really going to be talking about are conceptual problems that we have encountered. So we're actually not presenting solutions. We are presenting problems that we are trying to work through it. And so really in the next, 10 minutes, Zainab was really just going to try to go through a punch list of problems that we've had. And we hope that promotes a discussion about how to address these problems. So go ahead, Zainab.
Okay, can you hear me?
Yes.
Hello, everyone. First of all, as Marom said, I will be talking about all the challenges that we have been experiencing throughout these 2 or 3 years, as far as combining the computational head models with fMRI, which was and has been our motivation at the beginning. First of all, there is no need to emphasize that there are inter-individual variability is a key concern to the field, and there are several studies have been published that I just brought some of them. This is not all of those that has been using computational modeling to show that there is inter-individual variability as far as electric field induced in the brain in the targeted areas. And this is all for normal subjects. Of course, if you go to patient population, that's going to go to extreme, especially in the stroke and like atrophic subjects. So here, I'm just it's just an introduction to say that this is an example to show that the intensity and the electric field distribution for each montage and also across different subjects is different. And even if in the HD montages, that the whole promise is to reduce the distribution to a specific one that is focal, we still have these differences in the intensity produced in different brains across different individuals. So now back to our question that how we can quantify the electric field induced in the brain and regress it out in our experiments or in our clinical trials. Then this question comes up that what is the metric that you are going to use in order to quantify the electric field? There are several metrics that you can use. Here I just listed some of them, that it can be the peak of the electric field in that specific ROI, that is the targeted area. It can be the median, it can be just some percentage over a specific threshold that has been set in that study. And also, as electric field is a vector, we can assume the magnitude of that as a metric, or no, we can say the radial component or tangentials are important factors. So now we have a pool of metrics that we can pick from. And also another issue is we have been talking about ROIs, how you can choose the ROI that you want to quantify the electric field in that. So as I said, there are several other methods that you can use for choosing an ROI. Here we use two different methods just for the sake of comparison. The first one is just an atlas based, since our electrodes were on F3, F4 bilateral BFC. We wanted to segment all the areas that were exposed to electric field. So we segmented all the areas in the prefrontal regions based on the atlas in FSL. And also another idea was, why doing it functionally? Why not segmenting it based on the specific threshold of the electric field distribution? So we segmented the areas over a certain threshold that we knew that all these areas are getting good enough electric field. And then in the study, the question was, is there any correlation between the electric field in the areas and also the amount of activation in those regions or not? There's a lot of details in this slide, so for the interest of time, I won't go through all the details. But here we chose average electric field as a metric, and also this is the atlas-based segmentation. The study was sham-controlled crossover design. So as you can see, these blue dots are corresponding to sham sessions, and the red ones are the active ones. And yeah, the question was, is there any correlation between electric field and the activation after TDCS compared to before? And as you can see here, the frontal pole is the only area that has this nice correlation with electric field, while the sham session doesn't have any correlation. And I've shown it on the down right, that the frontal pole is the area that is getting majority of the electric field, which is obvious. It's like huge, and it's between the two electrodes. But this was not the only way we could have segmented areas. Another example of that was using it in the current flow drive segmentation. In this one, we did the same thing, but now, as you can see, there is no correlation in any of these areas, which is surprising. And I just, by bringing up all this challenge, I just wanted to say that we have chosen two different methods to select the ROI, and we ended up having two different results, which means that all these decisions of how you're quantifying that and where you're quantifying these things matters when you're doing the study. And
by the way, we also think if you chose your electric field differently. So instead of average, you choose median, you're going to get different stats. If you choose peak, you're going to get different stats. So these things, radial versus magnitude. So we haven't tested it, but we're strongly suspicious that depending on how you pick that as well, you're going to get to very divergent conclusions.
Yep, exactly. So another point that I wanted to make here is and by now acknowledging the fact that anatomical differences is a potential source of variability, and in all the clinical trials, we want to somehow take it out of the variation and explain why it is. There are two potential ways that you can do that. The one is the retrospective way, that everyone gets the same dose, let's say 1 milliamp for all the subjects, and after the study is done, we can go back and do the modeling and calculate how much electric field was each person receiving, and then calculate this as a regressor in the study. And also another way is like the prospective way, that each person can get a different current, which is based on the modeling is guaranteed to produce the same amount of electric field in the targeted area in the brain. Each of these approaches would have, I guess the first one is the easier one, but each of the approaches has their own underlying assumptions, which now we are going to talk about. The retrospective way to regress out the anatomical differences has these assumptions of the linearity, or at least monotonicity, and the polarity dependent of the dose response, meaning that if there is an increase in intensity in some subjects, there is an enhancement in the outcome. And in the prospective way, there is the only assumption that they're making is that the same amount of electric field will produce the same output in that ROI, which we don't know that yet because anatomical differences is not the only source of variability. But let's go back to the retrospective way, that the assumption is big. So based on what we have in the literature, I know this is a little bit complicated. Let me just zoom in. Based on the physics of TDCS, we know that there is this linearity that is dictated in the physics of TDCS. So we know that if you increase the amount of current delivered to the electrode, you would end up having increased electric field in the brain. So here I'm showing the anodal and cathodal of M1SO and HD electrodes, showing 1 milliamp and 2 milliamp for all of them. And if the 1 milliamp is producing X amount of electric field in the brain, doubling it to 2 milliamp would make the electric field in the brain area double. And this electric field is producing the membrane polarization in that areas. So based on what we have in the literature, in vitro slice of the animal models and also the in vitro of animal models, we know that there is this good evidence of linearity of the polarization based on the electric field. But of course, we know that those experiments the electric field is fully uniform, and also there is high intensity of the electric field in there. But still, at least linearity is somehow consistent here. But from the membrane polarization, if we go to human studies in the TDCS, then the story is going to be complex and different. So from the membrane polarization, let's say the electric field produced by TDCS is going to change the membrane polarization and then increase excitability, synaptic efficacy, and synaptic plasticity, and then cause enhanced network activity and ultimately cognition and behavior. But in the literature over these 2 decades, we don't have the evidence showing that increasing the intensity necessarily means enhancing the effects. Even so, not only we don't have that evidence, we have some evidence showing that that assumption doesn't hold, meaning that if you increase the electric field, the current intensity from 1 milliamp to 2 does not necessarily enhance the effects. So now there is this question of, is more current more or not? Based on what we have in the field right now, we cannot make any conclusion, and that's the assumptions of the linearity when we want to control for the effects of anatomical differences, it still is under question. So I just wanted to raise this question that this assumption is a huge one and we should be cautious about it. And another one that we were so interested from the very beginning was that we wanted to combine fMRI with TDCS and computational modeling. So the idea was we were doing pre and post. So we know that anatomical differences is not the only source of variability. We know that the state's dependency of TDCS is another important factor. So, so the question was how we are going to combine the state dependency of TDCS and also account for the anatomical differences. So we ended up asking this question with all the maps of the state of the brain before TDCS and a map of electric field during TDCS, asking this question that whether these two variables can explain the variability in the outcomes or not. And because it was a task-based one, we didn't go to the functional connectivity and all the other stuff. But of course, for the other imaging protocols, definitely there are different methods to look after those things. But another issue, I mean, that's the last one, but another issue that we came across was when we are comparing these two maps together, even like in ROI based or in a voxel based, the assumption that we are making is that each region is isolated and not connected, while we know that they're all connected and all the neighboring areas are exposed to electric fields. So probably this would elaborate that topic a little bit. That knowing the fact that these brain areas are interconnected and all the regions between the two electrodes are stimulated, which in our case it was, then we cannot go ahead and ask this question and not consider this fact that these areas are going to affect on the effect of the region of interest that I'm picking. So probably the solution would be not a definite solution, would be using some montages that are more focal, that at least we can control for the electric field in that specific area and be sure that some other ROIs that we know that has a correlation or connection with our region of interest are not exposed to electric field, or probably we can use some other analysis methods such as connectivity ones that we can account for the electric field distribution in each node and regress that out.
If what Zainab sounds a little bit overwhelming, because it was overwhelming to us, right? First, you're thinking, is the electric field in your ROI correlated with the change in the ROI? And we don't know that. And then on top of that, it could be 100% state dependent. So given electric field and a given ROI, what it does may be entirely dependent on the baseline state of that ROI, and we don't know that dependency. And then on top of that, most montages, like the M1SO montage, are stimulating most of the brain. And so now you have all your hundreds of ROIs, each with a different electric field, each with a different state, and each one communicating with each other ROI. And we really, got up to our necks sort of in this, and we don't have a proposal for how to simply address this, but we're just trying to show you the problem that we're struggling with. And when you're using a focal montage like the 4x1, you can remove some of these issues, but certainly not all of them.
Great, thank you very much, Marom and Zainab. It was a really nice presentation showing the challenges that we are facing in the field and the many things that we are supposed to think about in the next 30-40 years of our careers, so seeing how we might be able to do some interesting things inside. So we will have time in the kind of the last part of the webinar to discuss, so please keep your questions written and just we will discuss about those questions in details on how we might be able to kind of figure it out, how we might be able to collaborate. So let's move forward to the next speaker. The next speaker, Axel, Dr. Tischler from Technical University of Denmark. Axel, please. Let me just give you the screen so you will have the control for a screen. Just a second. Sorry, I'm simultaneously doing 10 different tasks together, so sometimes it just gets complicated.
Okay.
So, okay. Axel, you can share your screen right now. Can you?
I'll try. So now it should work, right?
Yes.
Wonderful. So, oh, sorry. Just let me get started here. Sorry for the hack here. So, I don't know how to get that now working. Oh, I think here. Would that be fine?
Yes, we are seeing that and we are seeing you and your voice really well. So that is good. That's great.
Okay, and you see my screen. So wonderful. So let's get started. So I was invited to talk about how computational dosimetry and MRI can inform each other. And I think I decided to not really talk about that because for two reasons. First of all, I think the best ways to combine the two things still have to be determined, and that's exactly what Marom was, for example, talking about. And the second reason is that there will be a second or another presenter, which is Daria Antonenko. She will give a recent example then on how we try to approach it as an example. So I thought it's also useful that one talk is more just focused on some hands-on methodological aspects that I think are just relevant to ensure a good quality for the comparisons. So I'll just briefly cover on four aspects here, namely both the tools which have been mentioned already by Marom, like ROAST or SimLabs. Well, they give you automatic segmentations, but actually the automatic segmentations, they really depend a lot on the input images, whether they're good or not. So I'll briefly cover that. I'll also talk about the electrode properties, which I think is really important also to take into account, and also to the positions. Then there will be some overlap in the sense that I'll talk about which measures to extract from the simulation results. So there will be some overlap with the talk from Marom, and I'll just round off with a few technical aspects. So let me first come to this thing about automatic segmentation. So what you see here is a brain segmented out from a T1 scan, of a typical T1 scan of a subject. And we scanned this person several times, so 3 times. So this is the first scan. And here we changed the parameters of the MR scanner a little bit. So as you should see more or less the same small change. And another small change, a third scan of the same subjects. Not big difference, right? I hope you agree on that. So, but we masked out here now the extra or non-brain tissue. So let's do the same thing again with the non-brain tissue. So this is the first scan. This is the second scan, and it's just a small change. It doesn't affect really the contrast in the brain, but it affects the contrast of the non-brain tissue. And this would be then the third scan. As you see, there's some apparent movement going on here, which is just an artifact. It's called fat shift. So the question is a little bit, how does that now affect really the segmentation accuracy of the automatic tools which we have? And so far we have SPM12 as the standard thing which we use to automatically segment it. So what we did here is, here on the left, you see the segmentation result from SPM12. When one of the T1 scans was actually combined with a T2 image, as shown here. So it's a multi-contrast segmentation, and here on the right side, what you see is the CT scan of the same subject. Yellow would be the compact bone, and the reddish here is the spongy bone. And in general, you see that there's a quite nice correspondence, I would say. So the quality is really nice. There's not much to do. So let's see how that changes when we only give a T1 as input. So this is the first T1 which we had. There you see already that part of the skull is missing. This is the second T1 which we had. There you see that more of the skull is missing. And the same subject again, the third scan, and you see another part of the skull is missing. So it seems to be a little bit stochastic. When you only have a T1 as input, what you get as the segmentation result for the skull out is just not robust. So how does it affect now the field simulations? So this is again simulation with the proper skull model, I would say. That would indicate that for this kind of unfocal montage, you have some stimulation here of the region which you intend to have, but you also have strong fields here in other regions. This changes quite a bit when you now have the one scan, which where the skull is segmented differently. So here you still have strong fields. In the target region, it changed a little bit, but also here in that region, it became really weaker. It changes even more for that scan, and then you can just go on with this game, and you can go back to this. So they're all changes, and they're just caused by that the segmentation is incorrect when you only had this T1 scan as input. And it's a little bit stochastic. So this is from one of our studies where we assessed this a little bit more systematically. In general, what we see is that when we have a T2 scan in the segmentation with SPM, then so here dice coefficient, it just means when it's higher, the better. And you see that it's for all different methods, it's relatively stable once you have a T2 scan in. And that changes really once you have only a T1 image as input, where some subjects really work out nicely. You have a quite accurate skull segmentation, versus for others, it's really failing quite a bit. So this just means that, to sum up that part, is that so far when you have head modeling and you base this head modeling on SPM12 segmentations, which both ROST and SimLabs do so far, you can't expect it to be robust for non-brain tissue. And it will just add a stochastic component to the field simulation results. And I think this will potentially also then inflate the inter-individual differences which we see. So as one thing to avoid that is that, well, I really recommend that you add a T2 scan to your imaging protocol, and that you also pilot your sequences, and obviously please check the quality of the automatic segmentation as a standard procedure and edit if needed. And if you do that, you should really expect some manual workload when you only have T1 images as input. I'd also like to mention that the images which I showed now were just actually of good quality, research scan quality. Can be really worse when you have clinical scans. So I think it's really relevant because otherwise we see a lot of inter-individual differences in the fields, but they are just coming from some stochastic performance of the automatic segmentation. Another thing I'd like to mention is, or go into, is the electrode properties and positions. So what is often used is still large PET electrodes, and I mean this very traditional image here shows it quite nicely. So when you add a large PET electrode on the skin, it doesn't mean that it really connects to the skin in a very homogeneous way, right? So you have strong varying contact impedances with some of the edges maybe not connecting at all, and some parts then really very well connected, and so on. So this is really adding some variability in how the current can enter the skin, and this variability is really hard to control for practically, and might actually contribute to the individual variability in the stimulation outcome, even though we don't really notice this because we don't have a good way of controlling for that. And obviously, when you simulate this, you normally simulate ideal electrodes, so you cannot really take that into account, meaning that also this is a potential source of differences between the real field which you have and the simulated fields. And for very large electrodes, what we also see is that, well, we not only have the effect that most currents try to flow through the edges, basically, but you also have that you have to connect to the electrode somewhere. So, and underneath the connector, there seems to be a stronger current entering the skin. And this is, again, some toy example here on the right, where what I did is just change the position of the connector of the red electrode from one time from the more inferior part to a superior position. It's shown here in the black positions. What you see here is that, well, one time the red region here, this region of the brain is really stimulated strongly, and here it's still stimulated strongly, but far less. For the return electrode, which is a large electrode, you see that this frontal region here is one time stimulated to a moderate extent and one time more or less not at all. So also this adds some variability, which is actually should be accounted for when aiming to really come to an accurate modeling. But I also I'm a little bit an advocate of not relying too much on large electrodes, because they're really hard to control and practice for. So one thing I'd like to bring up here is that, well, smaller circular electrodes with a connector in the center, as, for example, also used in 4x1 montages, give you much more control over where the currents really enter the skin, and will, by that, already reduce the variability, the inter-individual variability which you have, and also ensure that the current flow models will better predict the current flow. This obviously means that you also have to be accurate in the electrode positioning, because when you just slightly move the electrode, as shown by Alexander Opitz, this can already have quite substantial differences for the fields in the brain. So I think these are two practically relevant things I'd like to just bring up here. Then I'm going more into a topic which overlaps with what Maram said. So basically, what to report, what to extract. So one thing we are relatively sure about is that, well, it's probably cortical gray matter which is of most interest for us. But then, as said, it's both field strength, which might be interesting, but also the local field orientation relative to the neural target elements matter. So this is, in theory, all fine, but in practice, it's a little more difficult. So in TMS, it seems to be what we see so far, that we really can just rely on the field strength as to a first approximation, because the field strength is the dominant factor which really drives neural excitation. But even there are opposite hypotheses. Then for electric stimulation, it's thought that, well, that the normal field component, as already shown in the prior talk, polarizes the soma and also the dendritic trees of the pyramidal cells. So maybe we should really predominantly look at the normal field component. But it's just a hypothesis, right? So I think what we can do with this combination is really just take it from our explorative position and say, well, we should really, instead of trying to make strong conclusions in advance and on what we analyze, we should really, in a first round, try to see actually what parameters like field strength or normal or tangential components really give us the best fit to our fMRI results, and then try to explore in a second step why that is the case. That's just a toy example here to illustrate that. So these are two montages, as you see in the upper part here, a 4x1 and a bipolar montage, which I intentionally designed so that they give similar field strength distribution, because, but so both hit motor cortex relatively strongly. But obviously, as you might already expect, the current flow distributions are very, very different, right? So in the HD montage, we have currents entering consistently here in this area, the gray matter sheet, versus here we have currents entering on one side of the sulky and leaving on the other side of the sulky. So, and obviously we might think that this really makes a physiological difference. So one thing is that we can explore field strength as one parameter, but normal component might be also interesting, because it really gives us a very principled way of assessing the effects of the field orientation on the effects. So just to repeat what I said already before, just now with the normal component. So here in this region, we have consistently the field entering the brain across all the areas, versus here we have this nice red, blue, red, blue, red, blue pattern, meaning that the currents enter on one side and leave on the other side. So I'm not sure that we really should expect similar physiological effects here. So also the current direction might really be of relevance, and it's interesting to take this into account when we analyze data. And well, there's some evidence that the normal component really might be interesting also to look at. For example, here it was shown by Ilka Laakso that the electric field, the normal component, and the Joule crown here were explained best the inter-individual differences in MEP changes by TDCS. And in a similar way, also here it was that the normal component really changed a lot here in the area of the motor cortex, because the montage was changed, and this also had clear physiological differences. So all I want to advocate for is that basically field strength is so far shown quite often because it's easy to do, for example, and easy to understand. But I think also the relative is more difficult, as already pointed also out in the prior talk. So from a more practical point of view, I think, well, we want to do analysis in standard spaces, for example, in an I space. But I think what might be also quite interesting is to do analysis on the surfaces. Their FreeSurfer offers the FS average template. I think it has two advantages. First of all, it allows for quite accurate alignment of the individual cortical sheets. And then you have a quite natural way of defining and assessing normal and tangential field components. So also surface-based analysis might be quite interesting. Obviously, MNI is easier, I think, because it's really easy to combine it, straightforward to combine it with a standard SPM style of my analysis. Yeah, that's basically all I'd like to say here. And yeah, thanks for your attention.
Thank you very much, Axel. It was a really, really nice presentation, and it shows how how much devil we have in the details of those minor technical details that are going to be really important in terms of the question that we are exploring. That was great. So the next speaker would be Dario. So Dario would be following what Axel was talking about, and the data that they have collected and the results that they have. Dario, let me just give you the screen, and you can present what you have.
Okay.
Now you can use your screen.
Okay. Not sure. Is it working?
We can see it really well.
You can see the screen?
Yes.
Okay, great. Just do it. Okay, so thanks again for the invitation to present my work. I will talk about a recent study where we aim to link neurophysiological effects to electric fields. So in this study, we applied intrascalar TDCS and within subject design with three conditions: nodal, cathodal, and sham TDCS, and electrode montage was picked to modulate the activity of the sensory motor network. We measured resting state, fMRI during TDCS, and MRS before and afterwards. Resting state data was then used to quantify the coupling in the sensory motor network and the connectivity with the seat region. And MRS data was used to quantify GABA and glutamate. And in the end, as Axel also already mentioned, we included two additional structural sequences that were optimized and piloted before to give really good segmentations to do the computational head modeling and estimate the electric field distributions for each individual subject. Just briefly to give background on our initial idea, it was to improve understanding of TDCS, of course, as we all want, and we decided to use a setup where we kind of knew what to expect. So at least from previous study, we predicted to observe reduced or decreases in GABA and increases in sensory motor network strength. And so due to a nodal TDCS, basically, and in addition, we were curious whether we would observe a correlation between the individual decrease and increase on a neurophysiological level with individual induced electric fields modeled using computational head modeling. More specifically, I wondered whether we could quantify the quality of actual stimulation post-hoc, or in other words, maybe an OCD-like question we all have: has the PhD student done what he or she was supposed to do, or who else to blame for inter-individual variability of TDCS effects? So just briefly, on the neurophysiological level, we indeed found reduced GABA due to a nodal and cathodal TDCS, and reduced glutamate levels due to cathodal TDCS. Resting state connectivity in the sensory motor network was increased during a nodal and cathodal TDCS compared to sham. We also did a seed-based analysis with the seed in the precentral gyrus, which is underneath the active electrode, where we found increases in the left lateral and medial precentral gyrus and frontal clusters due to nodal TDCS, and increases in the bilateral medial, pre- and post-central, and occipital regions. So most importantly in this study, we did electric field simulation. We modeled the actual configuration of the electrodes individually for each subject and each condition, using photographs that were made during each session and rendering views then in the MRI cron software to delineate the position and really the orientation that was of the electrodes. Axel also mentioned this before, as the large sponsors in this classical montage do not touch the forehead of the subject. Nearly in any subject, actually, we measured the length of the electrode touching the subject's skin and used this for modeling. We used Synapse. I can only recommend the new version that Axel and his colleagues just released, and I'm happy that he gave a little bit introduction, so I can go to the results. As you can see here, the average and the variation of the electric field in our study sample. Maximum intensities are here observed in the frontal regions, including lateral, pre- and post-central gyri, with the high variability here. Looking at the normal component, which includes also the directional information, I also showed this, so whether the current goes into the cortex or out of the cortex. And we can see maximum values here in the central sulcus and also here in the precentral part in the left hemisphere. So we decided here to do a ROI approach, selecting 3 ROIs in the vicinity of the stimulated area. So one was around the mean coordinates of the MRS voxel here, and one was in the center of the head knob, and one was a so-called network cluster, which corresponds to the coordinates of the region where we found the strongest resting state connectivity effects. Comparing the electric field intensities, electric field strength was higher in the hand knob and the clusteroid compared to the MRS ROI, and normal components were highest. I cannot see my screen anymore. Were highest here in the cluster ROI compared to the both hand knob and the MRS ROI. So finally, these are the scatter plots showing the correlations between electric field strengths on the left here and the normal component on the right, with individual GABA level modulation and individual sensory motor network strength modulation due to a nodal TDCS. So exploring the linkage between these, we found that a significant positive correlation between the electric fields in the network cluster and the individual modulation of GABA. So the higher the electric field intensity, the higher the GABA reduction induced by a nodal TDCS, and likewise the higher the electric field intensity here in the normal component, the higher the increase in the sensory motor network strength induced by a nodal TDCS. So basically, we observed a link between individual current field intensities underneath the active electrode and neurophysiological effects of TDCS in this study. And most importantly, I think we show that there is a variability in induced electric fields that probably are related to the variability in neurophysiological effects. And just to show some example and go beyond the Roy-based variability, these are the distributions of the electric fields in each of the subjects that are included in the study, so from 1 to 24. And as I mentioned, we recorded the exact electrode positions here and orientations in each subject and modeled this for the anodal and for the cathodal condition. And you can already here see that when I go back and forth with the slides, that there is also an intra-individual variability. So not only that the fields are different despite the same aimed electrode montage between the subjects, there are also differences when we vary the electrodes from the anodal and cathodal condition in terms of the fields. Sorry. So if you pay attention here to subject 4, for example, but also you can see that between subjects there's a strong variability. Either the whole left hemisphere has high field intensities, and either it's a bilateral pattern or a more unilateral pattern. These are just some example data here when we use the rendering views, and then what this results produce of the electric field. So here you can see the nodal and the cathodal condition in the same subject. You see that here the electrode was a little bit higher. It may be due to a drift in the scanner. The subjects were like 2 hours, 2.5 hours in the scanner, maybe a little mispositioning in one or the other case. But the result is that we have a different distribution, which is more or less extreme depending on how it varies. But here you see the really small variation of the electrode position, but slightly different here. It's in the hand knob area, high intensities here, low intensities here, and also in other regions. So in my opinion, this points to the importance to interpret single or simulations based on single individual images with care, to include also structural sequences optimized for head model generation. This is what Axel described, to model individual configurations, which are ideally recorded also during the simulation setup. And if electric field intensities estimated by computational head modeling explain variability in TDCS effects, these could be then included as grouping variables, for example, or as a covariate, and maybe it is also possible then to predict effects on behavior or function. Yeah, this is my last slide, and thank you for your attention.
Thank you very much, Daria. It was a really, really nice presentation. Thank you. So we have time for a few questions, but as we are going to have discussions at the end, and we have just one remaining speaker. So that would be nice if we move forward for the next speaker, who is going to be Florian, and then we will move forward for basically discussion and asking questions from speakers. So Florian, let me just give you the screen. Just a moment. Okay, you can use the screen right now, Florian.
Okay. Can you see the screen already?
Really well.
Okay. Yeah. So what I'm going to show goes kind of into the similar direction of what we already heard from Daria. But in our case, since we are interested in transcranial alternating current stimulation, maybe we can look at it from a different stimulation modality. Yeah, so with TACS, we're trying to modulate brain oscillations by applying usually sinusoidal currents with the aim to synchronize endogenous brain oscillations. And for this synchronization, there is a lot of evidence from animal modeling. In humans, however, there's not so clear direct evidence for the mechanism. What we, however, see very often are these after effects, especially in the alpha band. They're pretty well documented and replicated several times. So we see an increase in power in the stimulated frequency band. And we have shown that this increase lasts for at least 1 hour when we apply TACS for 20 minutes. And we have, of course, similar concerns about the method in terms of sometimes effects do not replicate, effect sizes are small, and also very recently there could be a chance that the underlying mechanism of stimulation could actually be explained by peripheral effects. For example, stimulation of transcutaneous nerves or a flickering sensation that you have due to polarization of the retina. In terms of we have talked a lot about how much of the variability might be explained by individual anatomy. So our question was actually to test whether we can predict our TACS after effects in the alpha band from these individual simulations of the electric field. And here we did not take an RI approach, but we are rather splitting up these things into different compartments. So we have something like a precision aspect. How well does the electric field overlap with our with the source localized alpha oscillations? And on the other hand, we thought it would be a good idea to have a strength parameter. We're just seeing what is the maximum current that is arriving in the brain. And finally, since TACS is something that has frequency dependence, we also check whether a mismatch between the frequency that we're actually stimulating and the dominant frequency before stimulation also plays a role in modeling our effects. And as our statistical hypothesis, we first did a classic analysis where we just compared group means compared to a sham group. And then, of course, we tested whether we can predict our stimulation effect with the above model. We did not use fMRI or MRI. We were doing this study in the MEG, and we recorded 40 participants there. We did a little determination of their individual frequency on a 3-minute resting state recording before the actual main experiment. Then we recorded MEG for 10 minutes before stimulation. Then we applied stimulation for 20 minutes, and then we recorded post-MEG another 10 minutes thereafter. During that, participants had to do this very boring task that you see down there. Basically, they were staring at a fixation cross that would rotate approximately once in a minute, and they had to react with a button press there. That was just to make sure people remain attentive and especially keep their eyes open, because that can fundamentally change the alpha oscillations. Along with the MEG, we recorded a structural MRI, and analysis was done in the frequency domain, and these frequency domain data on the sensor level were then source projected using dynamic imaging of coherent sources beamformer, and we did individual simulation of the current flow for each participant using the ROSE toolbox. Of course, when you do these simulations, you get a rather complex results with many electric field values for each voxel in the brain, essentially. So we had to break this down a little bit to have actual measures of what I would call successful TACS targeting. As I've said already, there is something like a spatial component. So how well does the electric field overlap with the brain activity that we're targeting? And for that purpose, we computed a spatial correlation between the electric field that you could see at the down right with the source projected alpha activity that you can see down here. And so if they are very similar, there should be a voxel-wise correlation between the two. So if we have a high correlation, we have a good spatial precision. And the other question is, how strong are we stimulating? And for that purpose, we we search for the 10,000 voxels with the highest electric field magnitude inside the gray and white matter compartments, and fed that into our model. Also, as I said, TACS is a method where we have frequency dependence, and so we also included this frequency mismatch. I said earlier that we're estimating the individual alpha frequency beforehand, but as it turns out in several studies that we did, and there's we're often not, we still get some frequency mismatch. There are recent studies nicely showing that there are some drifts in alpha frequency with time on tasks, and also before the actual experiment, of course, we're not using the highest, most sophisticated approaches for artifact cleaning and signal cleaning, because we kind of have to be quick in estimating the frequency. So in many cases, there are mismatches between the individual frequency before stimulation and the frequency that we're using for stimulation. So we also included this factor. Now, if we have a first look at the individual electric fields, there is not much surprise. There is a lot of variability. On the right, you can see the fields for the different subjects, and I think with the blink of an eye, you can see that there is quite a lot of variability in terms of the field strength, but also there are differences in the topographies. And what is most important for us probably is to briefly characterize the correlation with our individual alpha topography. That was only moderate in this case, and also for some subjects. There's actually not much overlap between our stimulation, the electric field, and the topography of the alpha oscillation. If we then look at our traditional comparison of stimulation effects on the group level, we see a stronger increase in alpha power in the group that receives TACS as compared to sham. You can already see that in sensor level data. What was kind of surprising was that the effect was rather widespread and not really localized in occipital cortex as we would have expected. And also important, we do not see group differences in other frequency bands. If we look at the two stimulation groups, we do see that in both groups there is an increase in oscillatory power on the source level. Again, quite striking is that in the TACS group, it's a lot more widespread than in the sham group. Probably most interesting to you now is that we now use the power increase within these two clusters. We thought this is the most fair comparison because if we would have used a common cluster, probably we would have a little advantage in the TACS group. So in order to bias our response variable a little bit more in favor of the sham group, we decided to use individual clusters. So these light up areas that you see here for the two groups were used to extract the power for each of the groups. And now we used our we fed our predictors that I just explained into a regression analysis to predict the power increase within these two clusters. And what we see is we get a significant model here that explains quite a lot of variance. And we look at the single predictors, we see there's our measures of TACS targeting are explaining variability, but they're all interacting with the factor condition, which if you think about it, of course, makes sense because we have one group that did not receive a stimulation that should be effective in modulating brain oscillations. So we thought it's now, of course, most likely that the model only explains the data in the TACS group, which is why we see these interactions. So we fitted this model separately, kicked out the condition factor, and fitted the model separately for the TACS and the sham group. And as you would expect, we see that only in the TACS group, the model explains a quite huge proportion of the variance, while it is not able to explain data of the group not receiving effective stimulation. And here again, you see there are quite complex interactions that significantly predict our TACS effects between these different measures of TACS targeting. Since we have this huge amount of explained variance, we also did a leave one out cross-validation. So we fit the model based on 19 of these of our 20 participants in the TACS group and try to predict the remaining data point. And what we see there is our explained variance drops a little bit, but we're still explaining a solid amount of variance. So to sum this up a bit, we see that we can replicate previous effects that we saw after TACS in the alpha band. What was kind of surprising, and I think this is one of the few or studies that actually did source localization on the effect. So this is also something we should explore a bit more in the future, that these effects appear to be rather unfocal. What was quite striking is that individual differences in the electric fields together with the frequency mismatch explain quite a huge amount of variability in the power increase that we see, which stresses role of individual anatomy. But also what should be kept in mind, of course, when we compute these spatial correlations, there's also a chance, of course, that there are some participants that do not show such a clear alpha topography or kind of differ in terms of the spatial pattern of their alpha topography compared to other participants. For example, it seems like there are 4 to 5 subjects that show rather something that has motor areas involved, which could be something like dominant mood power, maybe. What is important is when I exclude these subjects from the analysis, still we're able to predict the power increase with our measures. And what I find quite fascinating about these things, what I thought might be interesting to share with you, is that what benefits we might be able to gain if we integrate our electric field modeling in statistical analysis. Because I think we're much closer to the actual underlying mechanisms, because we can feed in a dose-response relationship, which does not assume, as we do in a conventional t-test, that there is a consistent effect on more or less all participants, but we're taking into account that there are participants that receive non-optimal stimulation, and that therefore these subjects may not respond that well to stimulation or do not show any effects at all. So I tried to illustrate that a bit with the plot on the right. There might be the case that we have something like a moderate stimulation effect, or moderate differences between groups, or even non-significant differences between groups. But if we plug in some kind of dose-response relationship, we might see that the groups differ a lot in terms of these dose-response relationships, which might give us a more sensitive test for stimulation effects. And the other thing that I find interesting is, especially for TACS, where there is concerns that effects might be driven by peripheral mechanisms. It's rather difficult to explain how peripheral mechanisms could explain these dose-response relationships with our electric field models that are based on the current that is arriving inside the brain. And of course, after all, these results also emphasize that we might be able to increase the effects that we can see when we are individualizing stimulation parameters, montage, intensities, and maybe also be more adaptive or improve our estimation of individual frequencies in case of TACS. Yeah, and with that, I would like to thank you for your attention. If you're interested in some more details, there is a preprint available. Yeah, and I'm open for discussions.
Great, thank you, Florian. It's amazing. Being able to explain 52% of the variance is going to be huge. That is amazing. That was really nice. So now we have time for questions from speakers. Just to make things easier, those who are going to ask a question, they need to kind of open their video and just do something like shaking their hand. Just let me just be able to make sure that everybody would be able to ask their questions. So if there is any question from a speaker, from the first speaker to the last, the panel is open for discussion and for questions. Any question? Okay, Johannes.
Can you hear me?
Yes.
Yes.
Yeah, okay, cool. So thanks to all the speakers for those very impressive talks. Especially, I think, from Axel's talk, I got the impression that we have lots of variance in like the technical side of setting up our experiment. And as one of the things that he said was we should try to be more accurate in electrode positioning, I had the idea that for that we actually need like individual neural navigation or something like that. I would like to know from Axel how he thinks we could improve on that. And if I can add one more tiny thing, I would like to propose for the final for the consensus paper that we may try to figure out together what is the most optimal way of doing an TES experiment, and try to figure out what happens if we're not reaching that optimal state.
So maybe I just answer on the electrode positions. And I think for this specific audience, it's actually not so difficult because you do MRI. and you see the electrode or the gel at least on the MRIs. So I would just suggest that you actually set up a structural sequence which you acquire in addition to your functional MRI with the electrodes on the head. And it doesn't have to be the full structural sequence which you use for building your head model. You can also use a speeded up sequence with a lower resolution. You will still be able to accurately localize your electrodes on those. And I think that's actually then even more accurate than what you can reach with a neural navigation system. So I hope that answers your question a little bit.
Not really, because that's post hoc. That's what we can do after we've actually positioned the electrodes, right?
Okay. Yes.
While we're setting the electrodes on the subject's head, we need to have neural navigation or something like that.
Yeah, I agree on your point. So it's a good post-hoc control. So in terms of exploring what the effects of the field differences are on the outcome, there we can then take it into account. But in order to stratify the application, you would need a neural navigation system. Yeah.
Mm-hmm. And regarding the question of kind of electrode positioning, do we have any comment from other speakers or other colleagues in the room?
I had a quick follow-up question with regards to acquiring a structural scan with the electrodes on. Is there a problem with just acquiring your standard MP range with the electrodes on? Why would you need to acquire an additional structural sequence?
Yes. So just to be clear, MP range works. Additional elements, I recommend also to acquire an MP range without the electrodes on, because otherwise, when you reconstruct the head model, you will have just an artificial bump of the skin where your electrode has been. And this just is then making your head model a bit like incorrect, anatomically incorrect.
But do you think that would affect like your segmentation? I mean, surely it would just be, not taken as part of the tissue probability map.
No, I think everything which is just somehow on the head will interpret it by SPM at the moment, at least as belonging to the skin. So when you look at the head model, it will be just much thicker than there. So I think it's a good idea to really acquire the structural scan without the electrodes on.
It's just time.
But that's why I said the structural scan with the electrodes doesn't have to have the full quality. You can really scale it down so that it runs in 2 minutes or so, right?
Yeah, I was just thinking in terms of my scanner time. I was planning to acquire my structural with the electrodes on.
Yeah.
But you think it's going to be a serious problem for the reconstruction of the structural image or segmentation of the structural image?
You can test it out yourself. I mean, it might also depend a little bit on the electrodes which you're using. But what we saw so is that you can really nicely see them as skin bumps.
Oh yeah, I have. Yeah, I have seen them, but I just assumed that this wouldn't be a problem for when you were then, doing an analysis where you segment your brain to normalize.
Yeah.
Any other idea or comments regarding the electrode positioning and the discussion that we had right now?
I think
I would have a question for Axel.
Marum, please go ahead.
Just as far as the penalty of like gel smearing and other things, one interesting, if the gel has the same conductivity as skin, which is the case for things like TENS and other things that people are using, it potentially has zero impact on current flow, which is interesting, meaning you're kind of smearing it, because it's just like having more skin, right? You're just smearing it all over. Well, if you're using saline and that's dripping or drying out, the penalty is extreme. And so again, whether you're using pads with paste or whether you're using HD with paste, and so also as far as segmentation errors, it may be that if the automatic segmentation mistakes paste for skin, maybe the penalty isn't so severe. I don't know. I'm just putting that out there as an idea.
Yeah, just to add to that, I mean, what we saw is really extremely thick skin from classical pad or sponge montages, because sponge in water soaked gives a wonderful contrast and in UMR, and it's just then it's taking a skin, so then you suddenly have 1 centimeter more skin, and then you try to model your electrode on the top, and I think that's probably not a good. I think it really depends on the in your particular case, and you might want to just test it.
I would have a question to Axel. More general.
Agnes, before going to that, you are going to make a question, and Alexander, are you going to make a discussion about?
Yeah, I wanted to comment on the electrode accuracy. I mean, so we use like mirror navigation system. Also, if you have like repeat sessions, I want to make sure that you place those electrodes kind of at the same location that you had, like say a week before, 2 weeks. I think that is you can do with a neural navigation system really nicely. I mean, sometimes that's not always possible. So if you work with patients, don't have a lot of time, and sometimes you can do a photograph and try to reconstruct that. But also one thing that you can do is say you have like somewhat an uncertainty in the electrode location, so they can like just in the model maybe somewhat move the electrodes a little bit around and can see how strongly would your your modeling results change with say with that uncertainty in the location. Say okay, even if it's somewhat inaccurate, our electric field would not change so dramatically. You see, like, okay, this is a relatively robust simulation that will also give you some more confidence in those things.
Very nice, very nice. Any other comment and suggestion for the electrode? Okay, Yuran, please.
Just maybe a very small comment on this. So we've built together with EasyCap one cap where we mark the position of the electrodes, so that we can use it in a repeated manner. So I can maybe send around this picture of it.
Yurani, your question.
Yes, so it's a bit related also to the electrode positioning, and I'm mainly interested in using the head model for optimizing my simulation protocols. So not only in terms of where I want to put my electrodes, but also in terms of how much current I want to apply or something like that. So my question is more whether that's something that is already on the way to be implemented in SINITs, for example, or in ROAST, or if someone has done something similar already, or yeah.
I think it maybe yeah, Marom, I go first and then you answer for ROAST. I think it depends a little bit of what you want to do exactly. So I mean, when you want to keep the electrode positions fixed and just make sure that the same amount of current strength enters the brain, that you can do already now with the simulations, because I mean, it's a linear scaling between field strength and input current. So in order, you can just basically compare the fields which you then have simulated, let's say for 1 milliampere in the target area, and you see, okay, in this subject, it's now 50% lower than what I want. So I would then increase my current in reality by 50% to 1.5 milliampere. That would in theory equalize then the current strength in the brain. I say in theory because also the simulations have some uncertainty, and so that will work out to some extent, but not fully.
Agnes.
Marom,
do you want to add anything on that?
Yeah, I mean, I think Axel said it great. It's linear. So, I mean, there is an approach, obviously, where you pick an ROI and it prescribes any montage, right? And it gives you the montage to produce to optimize current delivery or focality for that. That's one goal. Another goal, like Axel just said, is just that you want to produce the same electric field in every subject. And then you don't necessarily need the optimization because things scale linearly. The one caveat to that I would point out is if you're using pads, then the peak is not under any pad, and the peak is actually moving, right? It's going to be different on every subject. And so when you're using large pads, it's a little bit more complicated. What are you normalizing to? Are you just picking one ROI and giving everyone the same electric field in that ROI, but it's completely wild outside those ROIs? ROIs. And you can in fact be increasing variability in other ROIs. And so that gets into the, so again, when you're using pads, I think that compromises what it even means to normalize to begin with.
Agnes.
Yeah, so great talks from Axel and Marom. I was just wondering, also as a suggestion to the consensus paper, when you model, or most of the modeling that has been done is more retrospectively, and you can tell the experimenter what probably worked well and what did not work well, and if there's some explanation of variability. But would you also like to use the experimental data to fine-tune the models? I mean, you already said there's some uncertainty in the models as such, and a lot of assumptions. Wouldn't it be worthwhile to prospectively have, for example, one subject where you know very well what the response, for example, to a certain type of TDCS is, to a nodal TDCS. There are some subjects like this where you always have this 1 milliamp, a certain increase in MEP size and so on. And you would systematically vary experimentally the strength of stimulation, also the electrode positions, assume that as a ground truth in a way, and then see what your model does with those variation, and then be willing kind of to to fine-tune the modeling. Is that possible, or is it not something that you think is feasible?
Could I go first again, and Mariam, you then add on, maybe? So I think in terms of model validation, I think there are two different aspects here. The one is really how well the models predict the current flow. And I wouldn't use fMRI or any physiological readout as my primary approach to test that. I would rather use direct recordings, intercranial recordings of the field as such, or methods like MREIT, which we're working on, because this is then a direct measurement of the quantity which I want to test.
Like a mechanical model in a way.
Yes. What we're doing is a current flow model, right? Mm-hmm. And that's it. And we want to just validate then basically that the fields are really equally close to the real ones. And then what we look at fMRI and also other physiological outcome measures is physiology. And there, as already Marom's initial talk very nicely shown, is that there are a lot of, or seen up basically, there are a lot of additional factors which also influence the variability. And it's also then how basically the fields really couple or change neural activity. So I think this is a much more complex scenario. And I wouldn't really like to try to tweak our models, which are current flow models, to explain that best. Because we can only make the current flow models accurate or not. And the next thing is then rather to understand how to best interpret the current flow and how to best use that information and to inform basically our physiological interpretation. Does it answer it a little bit? Yeah.
Yes, true. Maybe then you wouldn't actually change your model or fine-tune the model, but you could have like a systematic evaluation with the current flow that you predict, with the kind of known parameters and the known physiological response, and to see how this interacts, so that the experimenter has an idea how much your model with a injected field, for example, will actually tell him about about the physiology.
Yeah, and that's absolutely what is needed, I think. I just want to point out that from a strict point of view, a model validation using physiological data is just not possible, because the model predicts not physiological responses, but a physical entity. And I think this just is then two steps. We're talking about two steps here in a way. And both are really important to do, but we should maybe not mix them up.
Just, I don't know how much to add. I do want to, and obviously I think Florian gave a talk where he tries to get into linking, functional outcomes with current flow. We also have Dr. Harada here, and he's probably had done more work than anybody on individualized modeling and using that to explain data. But I think, Dr. Harada, most of your work has also been sort of retrospective and trying to correlate it. And your point is very good. There's been much less prospective data, and I haven't seen as much work on really trying to what Axel just said. These models that lack a functional, the current flow models lack a functional component. It just stops with electric field, and it has to be so much more complicated than that. And I think what you're suggesting is to you know, in a more humble way, just look at all the functional data that's coming out and correlate this functional data with current flow data and see if we can generate models that maybe then make functional predictions.
Yeah, more like neural networks, because those people that try to predict something, for example, stimulation within artificial neural networks, of course, they use the experimental data, but maybe it's not something you actually plan to do. You kind of want to stay with the electrical field injection and leave the neural networks to the next experimenter. Because otherwise, I would think you have to integrate the data from the empirical data if you really want to say something about the neural network level.
Sure, but what you're basically then suggesting is to add basically different modeling approaches together, which is interesting but also challenging because each of these have their own limitations and challenges. So it's very interesting, and it was a little bit brought up in Synapse talk already. But I think it will be a trial and error approach, I think, to a certain extent. So where we think we can try to do it our best way, then we will fail to a certain extent and learn from that. And it's really a learning experience what's going on at the moment, how to use these models best and how much they really predict in terms of technology.
And Alexander, are you going to add anything to that?
Yeah, I mean, I kind of picking up on those points, also model uncertainty, and I think in all this really nice talks that have been all pointed out, like those factors, and then, but one thing that hasn't really been stated so much is times of like accuracy of models predicting electric field strength, and you see all this nice correlation plot or regression line, but where you have on the x-axis have the electric field in any norm E, direction component, but basically what's not there is somewhat like an error bar of that estimate. And basically, I feel like if you add there like an error bar about the certainty of this predictive value, like your error bar would be longer than your x-axis. So like the error bar would probably be larger than the whole individual variability that you have between all your participants. And somehow, you can, questions like, what is like, say, the accuracy that you need in the model that such a correlation or like regression gives you a meaningful result is basically the error that is associated with, the individual field strengths,
But Alex, could we say, we can, as long as the rank order stays the same, we might be okay, right?
Yeah,
so if
no, exactly. But I would not. Yeah, if the rank order would be fine, but we also don't know that either. I would argue, right? I mean, if if you say the model predicts 0.3, the actual field strength might be like 0.1 to 0.5. That's what I would maybe say from our measurement results. And even if with that range, the rank order could still change, I think. So I think we're just not like fully there yet. As maybe something that you or Axel or others, maybe would be curious to hear their opinions.
There's been a lot of work on current flow validation and electric field validation, and I would say for all the unknowns, this field has been, that's been fleshed out over, a couple of decades now with some convergence. And there's a catastrophic lack of mapping that to functional changes, or asking how baseline brain state affects what electric field does, or network. So I'm kind of with you, Alex, that there's work to be done there, but it feels like there we're building on a foundation while going from these false color electric field maps to predictions of functional changes, never mind in a network. I feel like we are pretty ground level, there. Again, Florian, other people, we've heard work on this topic, but I don't feel we're so sophisticated there.
Dr. Hirata, do you want to add anything to that?
Yeah. So sorry, this is a conference place. It is so noisy. I do not hear everything. So basically, it is very easy to emphasize the uncertainty of current in the brain. But I think that as compared with other tissues, the brain conductivity is rather stable, at least reported until now. So it's just my feeling, but as compared with our result and other research group, basically in good agreement. So from that available data, maybe we have to try to compare with measured phenomena. And as already discussed, first one is model segmentation. Actually, I am basically the expert of human safety from electromagnetic field. Now I have been attending the conference. At the community, the same discussion actually there exists. The model segmentation itself, large error, maybe maybe 20%, 30% or more, then maybe we have to evaluate the electric field. If the electric conductivity is fixed, the error is not so large. It's just my feeling. Then the physiological response, it's actually the for TACS, it may depend on the frequency for synaptic effect. So all the components should be actually combined to discuss further for comparing with FMR. So it's actually my summary, and I'm unsure. Yeah.
Axel.
Yeah, I just want to add to that. So I think Alex certainly has a point, but I think also Marom has a point and that he says, okay, as long as the order effects are fine, we can start making these comparisons. And I think I agree that the computational models have been improved over the last decades. Well, maybe especially since we have more accurate numerical methods. What is still to be done, I think, is still work on segmentation, robustness. Then the other thing is we don't know the tissue conductivities with high certainty. That's another thing. And obviously, that's why I'm very interested in really physical measurements to get those better. But still, I think we're at a position where we can start making comparison with physiology, because that's what we want to do at the end. Just want to add one thing. I mean, order effects, as said by Marom, they stay the same across subjects as long as the subject group is quite homogeneous. I think when you have, for example, especially aging populations, the skull conductivity of very young and very old might really differ a lot. And suddenly there's a parameter in the model which we don't take into account so far. So I think this is just something to keep in mind. So as long as you have a homogeneous subject group, I'm relatively confident that order effects stay in place. When it gets more heterogeneous, I'm not so sure anymore. But that's interesting to explore, actually.
Any other comment or suggestion?
I'm very curious if if for any outcome measure, if you've got 10 subjects and you scan their functional state initially and their anatomical state initially, if anybody would be able to rank their sensitivity to an intervention, prospectively. And again, I know Dr. Hirata's worked on this, and we've heard stock from Florian doing this retrospectively, but that would be a very impressive piece of software where, again, you could take 10 people and do whatever you want to them, baseline measures, and then rank how much they would respond. And that, that's a high bar, for TDCS and for TACS. I would argue that does not exist for TMS and for ECT and for DBS also, where you can do it sort of so prospectively. But I wanted to put that, maybe someone can say I'm wrong and they can do this, but I'm not sure.
Alexander.
Yeah, I think then also in terms of like following up, what is like a number of participants that you need those predictions, and then you have all those, you know, all those choices of, you know, analysis you can do. You can do whole brain, you choose an ROI, you can choose like all those different field components. And so in some way, you maybe have to be more stringent with the statistics, somewhere coming like from the fMRI, you know, field, how that has evolved over time. Let's say, oh, you know, we choose like a small ROI, and then we find this correlation, and that's how we then build our paper and have a nice story. But in some way, just the degrees of freedom, or, you know, statistical models you can try are basically almost infinite. And I think there has to be then also be cautious to say, okay, I think for exploratory analysis, I think that's all great and fine. Say, okay, this has been exploratory, but in some way then also see, okay, can we then confirm or also replicate those studies, or can we pull that across a larger N? I think if you now do an fMRI study and have now your 10, 15 participants, you cannot publish that anymore in a decent journal. But like here in brain stimulation, still like they, okay, we do like 15, 20 you know, participants, and is to publish that in neuroimage or brain stimulation, for example. So I was wondering also, you know, what are your thoughts on that? What is like kind of minimum number of participants to have that such analysis actually also, you know, can make sense?
Bernard.
I guess we could think about implementing a resting scan at the beginning of all fMRI studies as like a collaborative effort. I was planning to do that for my upcoming study. It's just going to be 24 people, but I mean, if more people were doing that, then we could, you know, when we have similar montages, we could compare initial functional brain states as well as using the structural stuff.
Yeah, Mario, that definitely is something that we are thinking about in a type of kind of collaborative network that we already have, in terms of if there is any kind of potential that we can add something to the protocol that we are collecting, and being able to share the data together in the future, seeing how we might be able to start with. I mean, you have mentioned that it could be really simple, let's say resting state fMRI protocol that could be easily done in different lab, or be added to other studies that we are doing, and see how we might be able to use that data. And there are also kind of available data in the field that people have collected before, but they have never explored the, let's say, the computational head modeling part of that, and then we can kind of consider that as well. So that is something that I hope that we can do together in this network that we have. Bernard, do you have any question? You have any question? You want to?
Yes, thank you. Can you hear me, actually? Yes. Okay, thanks. So this is actually like a follow-up question, or maybe comment on what Axel was raising before, mentioning skull conductivity, changes in skull conductivity in aging populations, and that we had like to address this question also to realistically model electric field distributions. So I think we need a solution for that, and I think it still might be hard to resolve that based on MRI, but still it's one of the most important problems when you want to apply it to clinical populations, stroke patients, other kind of populations, because I think that's the target of most of us to actually apply it to the clinics. So what possibilities do you see, Axel, to take this into account and to apply it more to clinics?
Okay, so I think that's actually a quite ambitious goal already, not only to find out how much variability there is in aging, which is the first question, but then already how to take it in clinics. I think it needs a two-step approach, basically first to identify how large the changes really are and just report them. And that, I think, can be also a quite comprehensive study, because that doesn't have to be then geared towards a clinical implementation. The second thing is like a clinical implementation. Yes, I think there are things like EPT or EIT, which works with electrodes on the head, tries to measure the impedances of the tissues. I think those could be potentially adapted to give you individual conductivity estimates of the skull. But I think to have that really into a easy to use solution, it has to be really integrated in some commercial product, I think, so that you can really then roll it out in clinical applications. So at the moment, I think there's still lack of knowledge. How much does it really vary across the age range? There's a lack of methods how to assess it properly on an individual level. And then there's the implementation question, how to best bring it into a product or something like that. So I think it will still take a little bit of time.
Maram, do you want to have any kind of concluding note, anything that we kind of have as the last point?
No, I was just I was really surprised at how much convergence there was among all the speakers about the kind of issues that we need to work on and where the unknowns are. Everyone's saying, how do you correlate electric field with outcomes, and how do you consider individualization, and really Florian's point about ultimately increasing effect sizes, really, which is sort of the goal that we want to get to, either at the population level or at the individual level. So I for all the questions that were brought up, I really want to recognize how much consensus there is on the kind of problems we need to be focusing on.
Yep. Any other concluding notes from speakers? Axel, others, do you want to have any other last notes? Any other thing that people can like to add? Okay, great. So we are at the end of the second webinar. I hope that this webinar would help us to just get together and see what are the challenges that we have and how we might be able to work together to face those challenges in the next 10, 20 years ahead of us. And I hope that we can kind of find solutions in a collaborative way. So we will keep in touch, and we'll have the third webinar on September 26th. I will send you the details about the third webinar, and we will move forward together. Thank you very much for your attendance. I hope that you will have a really nice day.
Bye.
Bye-bye.
All right.
Bye-bye.
Goodbye.
Goodbye. Thank you.
Bye.
Bye-bye.
Bye, thanks.